Have you ever seen cool pictures of proteins? Things like this, with those cool like ribbons and sheets and cool colors and stuff, and wonder how the heck those were determined and how to interpret them? If so, the PDB is your friend, and hopefully this post will help you learn to use it better. So the PDB is the Protein Data Bank, and it allows you to see and play with in 3D different structures of proteins and protein DNA complexes like this, as well as just like DNA and RNA and all sorts of cool stuff. And so this structure and a lot of structures are solved using this method called X-ray crystallography. So more about this later, but the basic idea is that you get your protein or your complex to form this orderly, to like freeze in place in the same place throughout the crystal, and this thing we call a lattice. And then you beam X-rays at them, and these are really energetic light rays, and they're going to hit the molecules in that crystal, and then they get scattered. So they diffract. So they combine with one another, either constructively, so to build together, or destructively to interfere with the to like cancel each other out. And then the ones that build together, they're going to go and they're going to travel past the crystal and hit a diffractor, and then you get this pattern of spots. And then we work backwards from the spots to get this meshy thing, and then you work into the meshy thing, you build in that model. So the model is like what we were looking at before, where you had the so we were showing, it was shown in like cartoon representation, where you had the swirly things and the pleaty things. So we're going to talk more about all of that, and how we can use the protein database, so the PDB, to find these various structures that you might read about in papers or see pictures of, and actually explore them in more detail, as well as a kind of a guide on how to interpret the quality of the data and these various parameters like R value and resolution, class scores, outliers, all of this stuff. So I hope that this post will help people see that structural biology is something really cool. So let's start with what structural biology is. Different parts of different proteins will do different things, and if you just like the shapes of those different parts of the Swiss Army knife are specialized for doing those different things. And if you were to look at, say, the bottle opener part, you could see, oh, maybe that's used for opening a bottle. The same kind of thing goes for proteins and other macromolecules, where the shape and the function are intimately connected. And so we can't just look at a protein with our eyes like we would look at a Swiss Army knife, but we can use techniques like X-ray crystallography, cryo-electron microscopy, or cryo-EM, and NMR to get looks at the protein. And then we could do things like use site-directed mutagenesis, so make specific changes to parts of the protein and see how those affect the function, and then go back and forth between the function and the structure to try to learn more about each from the other. So one of the key, when we say that the structural biologists, when they use these methods like X-ray crystallography to figure out where the various atoms in a protein or DNA or other molecule are located, we say that they're solving the structure. And so that's what we mean. So, and by atoms, I just mean like those individual carbons, oxygens, hydrogens, nitrogens, etc. And so these scientists solve these structures, and then they deposit them in a database for everyone to see. And this database is called the Protein Data Bank, or the PDB. So this is mainly, it was established for X-ray crystallography data, but it also has cryo-EM data and some NMR data. And there are also more specialized data banks for those sources. And I want to show you today how you can use the PDB to look at and interpret structures. So I'm going to focus on crystal structures because that's what most of them are. And they can be kind of confusing to look at if you don't know what you're looking at. So let's quickly just review what a protein is and what we're trying to look at. So when you see pictures of proteins, you're normally seeing those things like at the bottom where you see that those arrows, we call those beta strands, and those are folded up into this beta sheet when you have the strands connected, and then you have these alpha helices, so those loopy things. And what you're seeing there is you're seeing the structural model. So that's basically a representation of where the atoms are located. And where these atoms are located is determined by the protein's underlying structure. So basically, proteins are these long chains of letters called amino acids that are linked together to form this long polypeptide chain. And then because the different amino acids have different properties, they cause the protein to fold up in this specific way to best accommodate all of those different properties. So specifically, the proteins have this generic backbone part, which kind of has this unique peptide bond backbone that restricts the motion of the backbone, which will come into play later, as well as those things that those R's sticking off. Those represent unique side chains. So you can think of it kind of like charms coming off of a charm bracelet. And so those are where you have different properties, and that's going to determine how the protein folds. Because they like to hang out in different places, and they also will restrict the motion of the backbone. So I'm telling you all of this because it's going to come into play later when we look at how we can assess a structure in the database. But for now, don't worry about that too much. But going back to what I said about those pictures at the bottom being models. So that's not what you will actually see when you solve a crystal structure. Instead, you get this kind of blurry map thing that's showing the positions of the electrons. So the electrons are one of the subatomic particles. So within an atom, you have amino acids are made up of atoms. So those are like your carbons, your oxygens, your hydrogens, and those atoms are made up of smaller parts. So in the center is the nucleus, where the positively charged protons and the neutral neutrons hang out. And then around that is an electron cloud. And so electrons are these negatively charged subatomic particles, and they're really tiny. But they whiz around in this electron cloud. And it's actually the electrons that are going to contribute to the signal that you see with X-ray crystallography. But when you make the models, what you're doing is you're trying to put like a pinpoint in the center, so like where the nucleus would be, but what you're actually seeing is the signal from the electrons. And so because electrons are in this like cloud, and because the detail isn't like the resolution you can get, so it's kind of like fuzzy, so what you end up is getting this meshy thing called an electron density map. But you don't get that electron density map either when you get your data. Instead, what you get is the diffraction pattern. So basically what you do with crystallography is you get the protein or whatever other molecule you want to look at, you get it to form this crystal, which is an orderly lattice. So basically it's kind of like a brick wall in all dimensions, where you have a basic repeating unit, and what's in that unit is the same, and you have a set of geometric rules like move up this far, move to the right this far, turn this far, and when you and then place a brick, do that again and then place the brick, do that again and then place a brick. So it's this repeating pattern where if you know what's within any one unit, then you know what's within the entire crystal. And the way that because of this unique symmetry, what happens is that what you get with the signal that you get is going to be representing basically like a single unit, but the signal's coming from the contributions of all these identical units. So what's happening is that the signal that I'm talking about is this diffraction signal. So the idea with crystallography is that you take this crystal and you stick it in front of an X-ray beam. And so X-ray is just really highly energetic light. So it's just like visible light, but it's more energetic. And what happens is when these X-ray beams hit the crystals, they're actually going to interact with the electrons that we were talking about before. And the electrons are going to scatter these X-rays. And so you have this beam of X-rays that's coming in one way, and then they're all getting scattered. And when the waves get scattered, what happens is that they kind of like run into one another. And you get something called constructive and destructive interference. So constructive interference is where the waves add together, and destructive interference is where they cancel out. And so what happens in a crystal is that the signal, because of this repeating pattern, what happens is that they cancel out almost everywhere except for specific points, and those points where you get the constructive interference, so the waves adding together, those bigger waves are going to go and they're going to hit a detector, and you get this series of spots called the diffraction pattern. Then what you do is you actually work backwards from that series of spots, from that diffraction pattern, to get the electron density map. So that weird blobby meshy thing we were talking about before. And that's going to show you the position of the electrons. And then you build the atomic model into that map. So that's where you you say, okay, we know that the electrons are in this area, and that the center of the atoms is going to be in the center of that, and then you can build the atomic model into the map. So how well you can do this is going to depend on things like the resolution of your data. So the resolution is like how well you can tell apart two different things. So with crystallography, if you have higher resolution, it's easier for you to see where to put the atoms into the map. And so the resolution that you collect is going to depend mostly on like the quality of the crystal. And so this is one of the things that you can find if you look in the PDB at an entry. So let's go ahead and look at an example. This is just an example I had found before. So it's looking at this protein called BCL11A, and it's this transcription factor that helps regulate when the fetal hemoglobin gene gets made. So basically, hemoglobin is the protein that carries oxygen in your bloodstream, and it's this multi-subunit protein, and one of the subunits, you make a different version of it. The fetus makes a different version of it that has like a tighter binding for oxygen, so that it's able to really snatch it out from the exhausted blood in the placenta. But then once you're born, you stop making that, and this BCL11A protein actually acts as kind of like the brakes to stop you from making it. And so some of the therapies for hemoglobin disorders like sickle cell anemia actually are taking advantage of this to try to get the adults to make this fetal hemoglobin to compensate for the defective hemoglobin caused by the disease. And so they were interested in looking at how this BCL11A protein binds to the promoter region in front of the fetal hemoglobin gene. So the region in front, how does it like put the brakes on this gene? And so they solved the structure of part of this protein. So here in this top, what you're looking at, so at the top they're showing where it binds, but then at the bottom where it says like BCL11A human, this is what we call a domain architecture. So basically proteins can usually contain multiple domains, and so we can mean different things when we say domains. Sometimes we're talking about, it's just basically like a part of the protein that we want to refer to specifically. So it's not, sometimes we can talk about like structural domains, where that often is like a part of the protein that'll fold independently of all the others. We can also talk about functional domains, so a part that does something. So a part that like like a DNA binding domain or something like that. So this is like a proline rich domain, glutamine rich domain. So those are just amino acids. And so those are just regions that have a lot of those. And so a domain is basically kind of like a shorthand for talking about a specific region. And it's also a lot of times defined, so those where you have that thin line in between like all those boxy domains, that's usually saying that that region is predicted to be like disordered or highly flexible. So disordered, highly flexible proteins don't for very good don't like to crystallize, because for crystallization they have to like freeze in place in the same place, and so flexible proteins aren't very good for that. So what they did was they only solved the structure of a part of this, and so they're showing you the structure here, but of course right here you're just getting like this snapshot of it. So say you want to look at it in 3D and you want to examine more closely how good the data is. So they're required to deposit that data into the PDB. And if you scroll to the bottom of the article, you'll see the atomic coordinates and structure factors were deposited in the protein data bank under the accession number 6K16. And as for the structure factors, that's kind of like the data from the data collection type of thing. It's not the raw data, but it's the kind of like the process data. And it's kind of hard to explain, but let's just look at what we can see if we go to the PDB. So you go to the PDB website and you enter 6K6. And here you find that. So you can see a number of different things here. So if we just look at the basic information, it'll tell us like the type of protein it is, the organism. So this is a Homo sapiens, so it's a human protein. It was expressed in E. coli, so basically they used bacteria to make the protein for them. Mutations, no. So this means they didn't introduce any mutations into the protein in order to get it to crystallize, other than like truncating the region. It tells you when it was deposited, when it was released. So usually they will deposit the structure before it's released, because they deposit it while like the article is under review and stuff, and then the PDB will hold it until they publish the paper, and then they can release it. You can release data without having a paper, but the authors usually, or the journals, I'm not sure how it works exactly, but they want to hold off on releasing the actual data until the paper is released. And then we'll get back to this part here, which is telling you about the quality of the structure. And then it gives you the information for the associated paper, as well as a list of what is in here. So you can see the molecule. So this is the B-cell lymphoma leukemia 11A. So that's the BCL 11A that we were talking about before. And it has these two chains, which we'll talk about. Those are just different copies of the same protein within this. And then the protein feature view, so this is the sequence of the protein. What you'll see here in gray, so these are unmodeled regions. So this region of the protein is going to show up in the model as like a dotted line, because it was present in the protein, but the data for that region wasn't strong enough for them to actually model it in. So when they got that blobby meshy thing, there wasn't enough of the mesh in that region wasn't high enough quality for them to actually try to fit in the atoms. You also see small molecules, so this is saying that it binds to, or that they also found a zinc atom in here. And then you get more information about the experimental stuff, so how they so this is an X-ray diffraction, so this is a crystallography structure. It's giving you information about these various crystallography terms that I'll get into in a minute. So let's get back to let's get back to, let's look at the 3D view. So if you go to the 3D view, this is where you can see the structure, and see it in 3D and play around with it. So right now, what you're looking at is you're looking at the a biological assembly, which is like the basic functional unit type of thing that you would think about if you were thinking about the protein. So let me go back for a minute, and just when I was talking about what a crystal is, and so basically a crystal, so we have when we talked about like the crystal being this repeating pattern of like brick sort of things, those brick sort of things would be your unit cells. Within the unit cell, there can be multiple copies of the protein or the complex or whatever it is that you want to look at. And the smallest unique part is called the asymmetric unit. And you can have multiple asymmetric units within your unit cell. And so we have these things like called space groups, which will let you make one unit cell from one asymmetric unit, and then lattice transitions where you make a whole crystal from one unit cell. So the important thing just to keep in mind is that you can use, as long as you know the asymmetric unit and you know the space group, you can figure out, and the lattice transitions, and you can figure out the, like where every single identical atom would be in every place throughout the crystal, and this will allow you to use that fancy software math stuff to figure out where the atoms were based on the signal spots you get in that diffraction pattern. And so what we were looking at when we were back at the PDB at that point, we were looking at one of these asymmetric units. So this is like the biologically relevant, one of these biological assemblies, which is one of the biologically relevant form. So in this case, it's the protein, so the which is in teal here, bound to the DNA region of that promoter sequence. So, but within the asymmetric unit, so remember the asymmetric unit was the smallest unique part. And so within that smallest unique part, we actually have two copies of the protein DNA complex. And so this is where we got that chain A and chain B. And so those are two copies of the same thing, but they're in like a slightly different position. So you can't like directly go from one to the other. They're in a slightly different, like the molecules are slightly different in their like position and shape or whatever. So you can't, so it's the whole, together they make the unique part. And then you have lots and lots of copies of them. So within this asymmetric unit, so we have the unit cell. So remember, that was where we can take that asymmetric unit. We use these space group formula, recipe things for figuring out how to make the unit cell from it. And then the unit cell with these lattice transitions can make the whole crystal. And so if you look at the unit cell, in this case, it has multiple copies of that asymmetric unit. And then you have multiple asymmetric units in the crystal. So right now we're looking at one of the biological assemblies. If we can choose what we're looking at. If we go here to the assembly ID, so if we wanted to look at the second one, you can see that this one is slightly different, and it has less of the protein is actually visible. So how did they determine what is visible or not? Oh, so you can also like look at symmetry. If I look at model, we see them both. So let's go back to the better looking one. Okay, so this was the chain. So you can see here this dotted line. That's a region that is unmodeled. So it was present in the protein, but the structure, like the data, wasn't good enough. And so you can see that such regions are shaded light gray in the sequence representation. And you can also, there's a lot of different things you can do to play around with various, seeing various things. But right now I want to show you the density. So if we look at the density, we'll just look at this part. And view, we'll do view around the whole structure. And update. Okay, so now what you see is you can see the structure that they built, the model that they built into this map. So if we can turn off this cartoon view, and we'll turn off these, and now you can see, so this is the sort of thing that you get, the electron map, the electron density map. So this is working backwards from those diffraction spots. You get this meshy mappy thing. And so if we look at this now, knowing what the structure is, you can see here, you can see like the DNA and that sort of thing. And then with the help of the software and knowing what the sequence is, then you can build in the model. And so it's nowadays it's software and stuff will help you do it. And you can see that it fits nicely in here. So when you do this though, you have to think, so like the data that you're getting from the diffraction pattern is what you're using to build this electron density map, and then you're building the model into the map. So you have a couple, because you're building the model into the map, there's going to be, you need to make sure that the model actually fits in the map that you're building. And so there's different values that we can use to assess how well the model fits the data. And then also there are things that we can use to talk about how good that data is. And there are also things that we can talk about how realistic the model is from like a structural and biochemical standpoint. And so the PDB helps us understand these. And so here they're showing it in these PDB validation sliders. And you can also get a lot more information if you look at the full report, but we're not going to go into that right now. But so one value that we talked about is the resolution. So that's basically how, that's talking about that meshy thing and how, basically how tight the meshy thing is around where the atoms actually are. And so if you have a big sausagey thing, that's poor resolution, and it's hard to place those atoms, and it's hard to tell apart what's what. But if you have a high resolution, then you can really start to see things like the side chains, so the part of the amino acids that stick off. And you can start to get like orientations and stuff. And so often sometimes the backbone is all that you're able to see, and all that. So sometimes you'll just model in the backbone. And it's harder to see the side chains, which tend to be more flexible, have weaker density, flop around more and stuff, and so are harder to model in unless you have higher resolution. So here we have a resolution of 2.5 angstrom, and angstrom is this unit for really small things. We also have this value, these R values. So these refer to how well the R free value is basically how well the data fits to your model. The R value with the work is kind of more, our work value is a little more complicated. Basically, when you're working with making the model, you want to make sure that you're not overfitting your model. So basically, it's kind of like that idea where if you have a curve, you don't want to, like if you were to use a quadratic formula or whatever, if you keep adding exponents, you can get the curve to fit your data a lot better. But it's not really generalizable because you're like fitting into the noise. And so we want to make sure that we're not fitting our model into the noise. And so what you do is you actually hold out some of the data and you only build your model with part of the data set. And then you test with the rest of the data set whether the model still works with that data. And so our work value is kind of like the stuff that you're working with. And so you can lower this value and so get a better fit of the model to the data. But when you then go and apply that model or that model to the data that you haven't worked with yet, so the data that you held out, that'll give you the R-free. And so you can see that the R-free is higher, which tells you that you're overfitting your model a bit. But your R-free value is always going to be higher than the R-work, but you want to make sure they're not too far apart. So you can see here the R-free is the top slider on these charts. So with these charts, they're showing you, so in the black line is the percentile relative to all X-ray structures. And then in the unshaded box, you have the percentile relative to X-ray structures of similar resolution. And then this is showing you where this model fits compared to those others. So we talked about the R-free, and so that's a measure of how well your model fits to the data. The clash score has to do with whether the atoms where you're, so now with the clash score, with the rest of this stuff, we're getting into the quality. So with the resolution, we got into the quality of the diffraction data. So the resolution is defined when you collect that evidence, and you can't change, you can't like improve that afterwards or anything. But then the R values are going to come for when you're doing the modeling phase, and they reflect how well the model fits the data. And then, so you could have a model that really well fits the data, but if it doesn't make sense, there's probably something wrong with it. And so when it comes to the making sense part, we're talking about like whether the atoms can actually like physically be where you think that where you say your model says that they should be. So one of the big things is steric clashing. So basically that's the idea that two molecules can't be in the same place at the same time. So you can't have, and that the molecules need their personal space. So basically you have this clash score where if the molecules, like if their electron clouds are too close or whatever, that it wouldn't be physically possible, then that'll be a ding on your clash score. Then we have outliers. So the clash score is kind of just based on where the atoms that you put in there are and how close they are to one another. But then the outliers are based on what is known about what positions like the atoms and like to take. And so this is often talking, so we can think about in terms of the protein backbone, which is represented with this Ramachandran outliers, and then the side chain outliers, and then the RSRZ outliers, which is kind of like whether things are sticking out of the map that you built in. So that's a little different. But with the Ramachandran outliers and the side chain outliers, you're looking at like the geometry of the bonds and the positions of the side chains. So basically a Ramachandran plot is looking at these like dihedral angles. So when we talked about peptides, amino acids being joined by these peptide bonds, and I said they were like special, they had the restricted motion. So there are only a couple places that they can rotate, and those couple places give you the psi and the phi angles. And there are specific places that those angles are restricted to specific like angle ranges. And so there are different places, if you were to plot where those angles were, so psi on the y-axis and phi on the x-axis, you can see you get this like heat mappy type thing of where the angles usually lie. And so you can see there's slightly different angles for beta strands and right-handed helices and left-handed helices, but there are these dark places where the angles are usually found. And if they're outside of that region, then we call them a Ramachandran outlier. And so this will differ for different amino acids. So like glycine can be more flexible because it's small. Proline has a more restricted range. And so when you have an outlier, that'll ding up on the Ramachandran plot score. So those are the basic factors that you can look at when it comes to the structural quality. If you go to the experiment tab, you can see more of the experimental details of the conditions for like the crystallization, the dimensions of the unit cell, the data collection, things about their resolution of more about the resolution of the data, and the software that they use to process it. You can also go to annotations to find out more about the various things that this protein is known to do. Then structure summary, we looked at that. Sequence, you can get more information, so about various, like where binding sites are located. So the gray, remember, those are our unmodeled regions. Secondary structures, so those are like alpha helices and beta sheets. So this yellow is saying that's an anti-parallel sense sheet. This pinkish red is a helix. So you can see that when you look at here, it'll like say, so residue, so that's the amino acid. You see position, and you see the author numbers. So you see those are different because this is just a portion of the protein. So the numbering, the PDB numbering starting from 0 is going to be different from the actual numbering in the presence of the full-length protein, which is where why you see like 40 versus 766, because if we remember back to the paper, we were only looking at that one region of the protein. We can also look at versions, so sometimes they will have multiple versions released, and so sometimes, like if you go back to the structure symmetry, it doesn't look like that's what happened here, but sometimes people will actually do like PDB redo, so they'll do a better make corrections to their model to help it better fit the data and better be more like geometrically ideal and stuff. So you might get different versions. You can also then download files. So for example, if you download the PDB file, that's going to allow you to open it in things like PyMol, which will allow you to make really high quality graphics figures for it. And if you download like the MMSIF or the structure factors and stuff, like, so you can actually see like the maps. So yeah, so you can actually fetch the structures directly from PyMol if you know what you're doing, using the same accession number that you used before. There's a ton of stuff on the PDB. So there's actually different versions of the PDB. So the PDB is like this worldwide consortium. So I've been showing you the RSCB protein data bank, which is the one based in the United States. But there are also PDB consortiums like around the world. So there's the PDB Japan, PDB Europe. Then there's also the like an NMR and cryo-EM, or not EM data banks too. And I really like the RCSB because they have a lot of really great educational tools, including some like paper models and stuff you can use. If you want to learn more about all of this, I highly suggest checking out their, I think it's the educational resources. If you go to the Guide to Understanding PDB Data, it's going to have a lot of very detailed information to help you understand what you're looking at. And so, sorry that got kind of long, and I don't know if it was really that helpful to people, but I hope it was, and I hope that this can help you appreciate what you're looking at when you see those pretty structural models. And so, happy exploring.